Hello students, in our previous session we have seen about Mendel's and his experiment. In today's session we are going to see deviations from Mendel's laws or experiments. Deviations means these are those findings which do not follow Mendel's laws. So let's get started with the incomplete dominance, codominance, multiple alleles and pleurotrophy. These deviations from Mendel's law is also called as neo-Mendelism. Before we get on to our neo-Mendelism, let us recall a few generalizations which were arrived by Mendel's on the basis of his experiments of garden pea plant, such as the first one. You have a single trait, a single gene is responsible for that inheritance of that trait, and you have two alleles for that trait. Now what do these two alleles do? These two alleles show interaction in which one is completely dominant. If you remember, with this we are talking about the law of dominance. But in neo-Mendelism, we come across a deviation, that is a single trait, a single gene. These two alleles show interactions which are not following the law of dominance, that is, they show a phenomenon called as codominance and incomplete dominance. Next observations of Mendel's experiments: the factors, that are genes for different traits present on different chromosomes, are sought independently. With passage of time, a number of deviations were observed, identified in the post-Mendelian era, that gave additional information on the patterns of inheritance. Now these deviations are described as neo-Mendelism. The next thing that was observed in neo-Mendelism was a single trait, a single gene, but more than two alleles. Remember, we had only two alleles in Mendel's experiment. Here you have more than two. This phenomenon is described as multiple allelism. Then you have the third observation, you have single trait but more than one genes. This is called as polygenic inheritance, showing differences in the interactions, or they are showing an additive effect. That is, for a single trait you have more than one genes, whereas in Mendel's we only had a single gene. The fourth observation, single gene influencing many traits. Now this is also new. This phenomenon is pleurotrophy. We had single trait and a single gene responsible, but here a single gene is giving you more than one or many traits. We will see these deviations in detail in this particular segment further. Students, now we will see the type of gene interactions. It was observed that the phenotypic expression of a gene can be modified or influenced by the other genes. These gene interactions are of two types. The first type is intragenic interaction. Occurs between the alleles of the same genes, and the example for this is incomplete dominance and codominance. It also occurs between the multiple allele series of a gene. So basically this interaction, intragenic interaction, is the alleles of a same gene. So you have one gene for a particular character and you have its alleles. So the interactions between the alleles of the same gene. Then the second kind of interaction is intergenic interactions. This is a non-allelic interactions. Why? Because it occurs between the alleles of different genes present on the same or different chromosomes. That is, you have alleles that are interacting which are not having this or do not belong to the same gene. They belong to different genes and they are also present at different chromosomes. Example, pleurotrophy, polygenes, supplementary, complementary genes, epistasis. So some of these interactions we will learn in this chapter. The first intragenic interaction that we are going to see is incomplete dominance. Now what happens in this kind of dominance? In the incomplete dominance, both the alleles, that is the genes of an allelomorphic pair, express themselves partially. one allele or the gene cannot suppress the expression of the other allele or the gene completely. In such cases, there is an intermediate expression in the F1 hybrid. Remember the law of dominance? You have one allele which is completely dominant over the another. But here you see one is not able to be completely dominant over the another, and hence there is partial expressions of both the alleles. A well-known example is the flower color of mirabilis. If a red flower, that is capital R's plant, is crossed with a white flower, that is 2 small r plant, then the F1 offspring has a pink flower instead of a red flower. Though the genotype comes to be of capital R and small r, but the phenotype is different. Here, have a look at the diagrammatic representation here. The parental generation, I take the red flower, the red being the dominant trait, and the white flower, the white being the recessive trait. When the gametes are formed in your F1 generation, as per the law of dominance, this flower should have appeared red. But in this particular case, the flower is appearing pink in color. So this is an intermediate expression. But again, there is no mixing of these alleles. So when you come to the second generation, that is the F2 generation, on selfing your F1, you get your recessive trait back. But this particular observation is a new observation which is not seen by the Mendel's experiment. In this particular case, you will see the genotypic ratio remains the same like Mendel's, that is 1:2:1. But in this case, the phenotypic ratio changes. Instead of 3:1, we have the phenotypic ratio same as the genotypic ratio, that is 1:2 pink, making it 2 and 1 white. So 1 red, 2 pink and 1 white, giving you the ratio, phenotypic ratio same as your genotypic ratio. The next intragenic interaction, student, is codominance. Now in codominance, both the alleles, that is the genes of an allelomorphic pair, express themselves equally in F1 hybrid. Such alleles which are able to express themselves equally independently in hybrid are called as codominant. Codominant means both are equally dominant. Neither is one allele suppressing the expression of other allele, or neither are both expressing themselves partially. Both are are getting expressed in the F1 hybrid, and hence the term codominance. So thus you can conclude in codominance both the alleles are expressed. None of it is suppressed as per our law of dominance. Here I have shown you a few examples of animals showing dominance. Here you can see the flower. You have a red and a white petal, a pink and a white petal, a red and a white petal, which means that in the parental generation, one flower was red in color and the other was white. Here in this case, one flower was pink in color, the other was white. I've shown you an example of codominance in cattle. Here you have a white cow and you have a red cow. Now in this case, when both are coming together, neither of the trait is being suppressed. You have the F1 generation, which is called as roan, which has both white and red or the brown patches on it. I've given you the diagrammatic representation of the cross. Here the parental generation, you have the red and you have the white. So you will see none of them are shown as recessive because both are equally getting expressed. So both have been denoted with the dominant letters, that is the capital letters. In the F1, when both the gamete formation takes place, results in the F1 generation, it has both red and white patches. But again, when you self them in the F2 generation, you get one red, two roans, and you get the white trait back. So here, if you put that in a Punnett square, again, you will see that the genotypic ratio is that of Mendel's law of dominance, that is 1:2:1. But again, the phenotypic ratio here is altered, and instead of 3:1, you get 1:2:1. Students, the next genic interactions that we are going to see is multiple alleles. More than two alleles or alternative forms of alleles of a gene in a population occupying the same locus on a chromosome or its homologue are known as multiple alleles. Now, multiple alleles arise by mutations of a wild type of a gene. A gene can mutate several times, producing a series of alternative expressions. Now, different alleles in a series show dominant recessive relationships or may show codominance or incomplete dominance among themselves. The wild type is dominant over all other mutated alleles. Have a look at this chart here. It shows the human blood group. Here, see the alleles that you have, IA, IB and I. So you have three different alleles instead of two. Now when IA and IA, two IA come together, the blood group is A. IA and IB coming together, the blood group is AB. And IA and a small I coming together, you have the blood group A. Then you have again IB or IA coming together, again the blood group remains AB. You have IB and IB coming together, you have the blood group B. IB and small I coming together, the blood group is still B. And if only you have the two I's coming together, the blood group is O. So in this particular case, you can see instead of two alleles for a particular trait, that is the trait of your blood group, you have three different alleles responsible for the inheritance of your blood group. And again, they show different kinds of interactions. For example, IA is a completely dominant over I. IB is also completely dominant over I. But if you see IA and IB, both are equally expressed and they show the phenomenon of codominance. So these are called as multiple alleles. Simple, more than two alleles for a particular trait. Students, the next interaction is pleurotrophy. When a single gene controls two or more different traits, it is called pleurotrophic gene, and the phenomenon is called as pleurotrophy or pleurotrophism. So you have a single gene and more than one trait or more than one character that the gene controls. The phenotypic ratio is 1:2 instead of 3:1 because of the death of the recessive homozygous. The disease sickle cell anemia is caused by a gene which is called as HBS. Now a normal or a healthy gene is HB capital A and it's dominant. The carriers, that is the heterozygous condition, will have HBA and HBS. This is the one responsible for the sickle cell trait. They show signs of mild anemia as their RBCs become sickle shape, that is half moon shape, only under abnormally low O2 concentrations. The homozygous which receive with the recessive genes HBS, however, dies of total anemia. Thus the gene for sickle cell anemia is lethal, that is, it leads to the death of the bearer in homozygous condition and produces a sickle cell trait in the heterozygous carriers. So here you have two different expressions that is produced by a single gene, which is HBS. Students, have a look at the cross here. You have the parents, the phenotype, you have the sickle cell carrier and the male sickle cell carrier and the female sickle cell carriers. So the genotype is going to be HBA and HBS in both the cases. So at the time of gamete formations, they are going to separate, and the offspring genotype is going to be when you have HBA and HBA coming from the both the parents, that offspring is going to be normal. If you have HBA and HBS coming together, then you will have them as sickle cell carriers. And if you have both the HBS coming together, then you will have the sickle cell anemias, and this particular individual will die, giving us the ratio of 2:1. Here you can have a look at this image here. This is your normal structure of your normal hemoglobin, and this is the structure of the sickle red blood cell. So this can show you this particular gene, which is HBS, is also called as a lethal gene, which is responsible for the death of the bearer, or the one who has both these genes and is homozygous for this particular trait will die. And because of this reasons, marriages are in between sickle cell carriers is always discouraged. Thank you, students. Take care, be safe, and keep learning.